Welcome students. In this session, we will be covering up the topic reproduction in bacteria, vegetative and asexual. I am Ms. Karen Lenisha Cabral. Now, coming to the outline, we will be dealing with the vegetative reproduction in bacteria, that is binary fission, and asexual reproduction in bacteria, that is endospore formation. What we will learn in this session is the process of binary fission in bacteria, what exactly is an endospore, and how are endospores formed. Now, when we talk of bacteria, bacteria are found everywhere. They are present in humans, they are present in various conditions, environmental conditions, may it be heaty, or may it be in snows. Now, how exactly do they reproduce? Now, here bacteria reproduce in three basic forms. That is, one is vegetative, asexual, and sexual form. In this module, I will be dealing with the vegetative and asexual reproduction. So, under vegetative, we have binary fission, while in asexual reproduction, we have endospore formation. What exactly are these? We are going to see next. Coming to the first one, that is the vegetative reproduction, we have binary fission. Now we have prokaryotes as well as eukaryotes. Now prokaryotes are simple form of organisms. That is, when we say prokaryotes, the first thing that comes to our mind is bacteria. Whereas when we say eukaryotes, they are complex organisms. Like humans, we belong to eukaryotes. But there are also other organisms which are of simpler forms which belong to eukaryotes. There are organisms which do show binary fission. Now, when you say binary fission, it is the most simplest form of cell division, wherein you will have the mother cell or the parent cell, which will divide into two identical daughter cell, wherein if you look at the genetic makeup, it will be exactly the same. Now, when we say binary fission, it is nothing but you will have the cell which divides in two equal halves. And when you say equal half, the first step is it's not just the cell, but the most important part is the genetic material, that is the DNA of it. So if you look into the first image right above, you have the mother cell, and then you have within it in the center, you have the DNA, that is the chromosome, which is located in the center. Now, what will happen to the double-stranded chromosome? It will unwind itself at the origin, so it will form a template, and a complementary strand will be made for each strand, for each template strand. Ultimately, you will have two double-stranded DNA. If you look into the second image, you have two DNA that are formed. So as this DNA is formed, the cell elongates. That is, the plasma membrane and the mother cell starts elongating. Now, why is this required? Because now the two DNA will move towards the two poles. Once this is achieved, there will be an invagination into the cytoplasm, because now what needs to happen is there needs to be formation of a septum, that is a separation, a wall between the two, so that they can form into two individual identical daughter cells. So right in the center you have the formation of a septum with a protein, that is FTSZ ring, wherein these proteins are going to get accumulated in the center, wherein a wall layer will be formed, and ultimately you will have the separation of the two. So right at the end you will have the wall layer, that is some amount of plasma membrane will be shifted to one pole, some amount of plasma membrane will be shifted to the other one, along with the DNA of that was replicated before. So ultimately in the first one, you have from one cell you have two daughter cells which are formed, and this is the most simplest form of division. Coming to asexual reproduction, we talk of endospore formation. Now, what exactly is endospore? Now, bacteria form dormant structures which are called as endospores. Now, when are these structures formed? During harsh conditions, that is under certain stress conditions. That could be physical stress or that could be chemical stress, maybe due to drought condition or due to UV radiation or disinfectant. When this kind of bacteria experiences these conditions, the harsh conditions, what it will do is it will go in its dormant phase, wherein the cell will become inactive. So, but it won't be dead. once it achieves or once it finds a favorable condition, it will start germinating again. So this is the best way for a bacteria to protect itself and not just get extinct or kind of die, but it is going to remain in its dormant phase. Now endospores may be centrally located, or they could be subterminal or terminal. This is within the bacterial cell. Now an endospore is formed within a vegetative cell. Looking at the structure of an endospore, you have 5 structures that make up an endospore. First one is the exosporium. You have the spore coat, you have the cortex, then you have the core cell wall, and you have the nucleic or nucleoid or the genetic material. Now if you look at an endospore, it looks something like this. So we have all the 5 components. Now exosporium is the outer layer, which is pretty thin and it is delicate. So it is the most delicate layer that is formed on the outer side. Then you have the next layer, that is your spore coat. Now inner to that you have the spore coat, which is a much thicker layer, and it is made up of layers of proteins. This layer protects the endospore from all harsh conditions. This layer makes it resistant to all other conditions, environmental conditions. Then inner to it you have the cortex. Now if you look over here, the cortex is made up of many layers. So it is pretty much thick. So it is the thickest layer, and it is made up of peptidoglycan. And inner to that you have the core wall or core cell wall. Now what is the function of it? It basically protects the nucleoid. So the nucleoid is well protected with this core wall. And right in the center you have the nucleoid, which has the histones along with its genetic material. Now coming to the formation of endospore, how exactly it is formed. The process of formation of endospore is known as sporulation or sporogenesis. And it is a very complex process, but it has 7 basic steps. First one is the formation of axial filament. Then you have the formation of septa. Third one is engulfment of a 4-spore. Fourth one, you have the formation of cortex. Then the 5th step is the formation of protein coat, followed by spore maturation and enzymatic degradation. Now all the steps that I've just mentioned are in this particular illustration. Okay, the first thing what happens is, this is a bacterial cell. So the first step being formation of the axial filament. So right now over here we have the DNA that is placed centrally within the cell. So first what needs to happen is, the DNA needs to replicate. So there will be replication of this genetic material, and it is bidirectional. So you will have genetic material wherein it is going to divide. And then what will happen is, you have the formation of septa, wherein if you look into the second diagram, you will have a wall layer that is formed. Now once this wall layer is formed, some amount of DNA gets transferred on one side, and the cell that is formed, the wall layer, one part of the cell will be smaller as compared to the other one. Now in the third step you have engulfment of the 4-spore. Now what happens, 4-spore, or it is also called as a pre-spore. Now what happens over here, the mother cell extends its membrane and covers this immature spore, allowing it to grow. So the mother cell extends, ultimately forming 2 layers of plasma membrane, and in between you have the formation of an inner space. Then in the 4th step you have formation of the cortex. Now within this space, that is the inner membrane space, you have the formation of the cortex, that is you'll have layers of peptidoglycan that is going to get accumulated between these steps. And then what will happen is, once you have that, then you have the formation of the protein coat or a spore coat. So if you see in this diagram, you have the spore coat which is formed. You have the spore coat which is formed over here, so which will actually help the spore against any drought condition or any physical or chemical condition that it is going to face. So ultimately it is protecting, the bacteria is protecting its genetic material. Even if under harsh condition, if the mother cell happens to get dehydrated or die out, the endospore will be in its dormant phase. And at the end, you have the last step, you have enzymatic destruction. wherein you will have the enzymes which is going to destroy, and ultimately you have the new spore, the endospore that is formed, which will be released through destruction. So upon a favorable condition, this endospore will start germinating again by activation and germination. Ultimately, this free spore is going to be released into the environment through wind, through various factors, and it can travel into various places. Now, if the condition is favorable, a bacteria can reproduce within 20 minutes. Thank you.